Welcome to ABC tutorial. Today we'll be talking about the cardiovascular system. The cardiovascular system consists of the blood, the heart, and blood vessels. The heart is a relatively small organ, about the size of a closed fist, weighing about 250 to 300 grams. It is located in the mediastinum, with 2/3 of its mass to the left of the midline. It has a base, an apex, and 3 surfaces: the left, right, and anterior surfaces. It is more like a cone resting on its side. The heart is surrounded by a protective membrane known as the pericardium. The pericardium are of 2 types: the outermost fibrous pericardium and the inner double-layered serous pericardium. The 2 layers of the serous pericardium are the outer parietal and the inner visceral layers. The parietal layer is fused with the fibrous pericardium, while the inner visceral layer adheres tightly with the heart. This visceral layer of the serous pericardium is also known as the epicardium. The space between the parietal and visceral layers of the serous pericardium is known as the pericardial cavity and usually contains thin film of lubricating fluid known as the pericardial fluid. The heart wall is made up of 3 layers: the outermost epicardium, the middle myocardium, and the innermost endocardium. The epicardium is made up of the visceral layer of the serous pericardium, adipose tissues, blood vessels, and lymphatics that supply the heart. The myocardium, which is the muscular layer of the heart, makes up approximately 95% of the heart wall and is responsible for the pumping action of the heart. The innermost endocardium lines the inner surfaces of the heart chambers. The heart is made up of 4 chambers: the 2 superior receiving chambers, which are the right atrium and the left atrium, and the 2 inferior pumping chambers, the right ventricle and the left ventricle. The coronary sulcus marks the external boundaries between the atria and the ventricles, while the anterior and posterior interventricular sulci marks the external boundaries between the right and left ventricles. The right atrium receives blood from the superior and inferior vena cava and also from the coronary sinus. The right atrium is separated from the left atrium by the interatrial septum, and blood passes from the right atrium to the right ventricle through the tricuspid valve. The right ventricle forms the anterior surface of the heart and is separated from the left ventricle by the interventricular septum. Blood pumped by the right ventricle passes through the pulmonary valve into a large artery known as the pulmonary trunk, which divides into the right and left pulmonary arteries and carries blood to the lungs for oxygenation. The left atrium receives oxygenated blood coming from the lungs via the 4 pulmonary veins. The left atrium delivers this blood to the left ventricle via the mitral valve. The left ventricle, which is the thickest of the 4 chambers, pumped oxygenated blood through the aortic valve into the aorta and to the body systems. Some of the blood from the ascending aorta flows into the right and left coronary arteries to supply the heart wall. After supplying the heart wall, the coronary capillaries then form the great, middle, small, and anterior cardiac veins, which coalesce to form coronary sinus to drain into the right atrium. Cardiac muscle cells are branched and attached to the neighboring cardiac muscle cells by intercalated discs, which contain desmosomes and gap junctions. The gap junctions allow action potential to conduct from one cardiac myocyte to another and allow the entire myocardium to contract as a single coordinated unit. The cardiac myocytes contain a large number of mitochondria. They also have actin and myosin filaments and sarcoplasmic reticulum, just like skeletal muscle cells. Some specialized cardiac muscle fibers have lost the ability to contract and specialize in generation of action potentials for the entire cardiac muscles. These cells are known as autorhythmic fibers and are self-excitable and repeatedly generate action potentials that trigger heart contractions. Because of these specialized cells, the heart muscles don't need impulse from the nervous system to contract, a reason why the heart may still continue to beat after being taken out from the body. These cardiac conduction systems include the sinoatrial node or SA node, the atrioventricular node or AV node, the atrioventricular bundle of His, the bundle branches, and the Purkinje fibers. The sinoatrial node is located in the right atrial wall. It typically acts as the pacemaker, setting the rhythm for the contractions of the heart. The SA node typically initiates an action potential at a rate of about 100 times per minute. By conducting along the atrial muscle fibers, the action potentials generated by the SA node reaches the atrioventricular node or AV node, which are located around the opening of the coronary sinus. At the AV node, the action potential slows down considerably, providing time for the atrial contraction to empty their blood into the ventricles. From the AV node, the action potential enters the atrioventricular bundle of ease, from where it enters both the right and left bundle branches, and finally to the Purkinje fibers. The Purkinje fibers transmit the action potential to the ventricular muscle cells, causing their contractions and pumping of blood through the outflow tracts. The cardiac conduction system and the cardiac myocyte exhibit different forms of action potentials. A resting cell is said to be polarized as the inside of the cell is more negative than the outside. In a resting cell, there is more sodium and calcium outside of the cells than inside, while there is more potassium inside of the cells. These differences in ion concentrations across the membrane are maintained by various pumps that pump sodium and calcium outside of the cell and potassium inside the cell. The action potentials are as a result of opening and closing of voltage-gated ion channels on the membrane of the cells, leading to depolarization and polarization of membrane potentials. Depolarization occurs when the inside of the cell becomes more positive, usually as a result of influx of positive ions, while repolarization is used when the inside of the cells becomes more negative, usually as a result of efflux of positive ions. The pacemaker cell membrane has three types of ion channels: the funny channels, the slow calcium channels, and the potassium channels. In the pacemaker cells, the resting membrane potential is about -60 mV, and there is a presence of funny channels in the pacemaker cell, which is open at the resting membrane potential. These funny channels allow sodium to slowly drift inside of the cell, leading to depolarization known as the pacemaker potential. This slowly brings the membrane potential to -40 mV, at which stage the calcium channel opens, leading to influx of calcium ions into the cells, leading to further depolarization to about +10 mV. At the peak of depolarization, the potassium channel opens, leading to efflux of potassium ions, causing the inside of the cells to become repolarized. The original ionic gradients are maintained by the ionic pumps and the cycle starts all over again. The three voltage-gated ion channels, majorly responsible for the action potentials in the cardiac myocytes, are the fast sodium channel, the slow calcium channel, and the potassium channel. In the cardiac myocytes, the resting membrane potential is about -90 mV, at which stage all the ion channels are closed. Action potentials in the cardiac myocytes start when the action potentials in the neighboring cells cause sodium and calcium to diffuse through the gap junctions, leading to slight depolarization of the cell to about -70 mV. At this stage, the fast sodium channel opens, leading to rapid influx of sodium and a sharp depolarization of the cells. During this time, the slow type calcium channel also opens, leading to slow influx of calcium ions. At the peak of depolarization, the sodium channel closes quickly and the potassium channel opens, leading to a slight decrease in membrane potential known as early repolarization. The calcium channel, however, remains open and the potassium efflux is balanced by the calcium influx, leading to a plateau. This calcium influx also triggers calcium release from the sarcoplasmic reticulum, responsible for the contractions of the cardiac myocytes. Finally, the calcium channel closes, leading to repolarization of the cell by the efflux of the potassium ions, thus completing the action potential cycle. The original ionic gradients are maintained by the ionic pumps, and the action potential can then start all over again. Contraction of the heart is known as systole, while relaxation is known as diastole. As action potentials propagate through the heart, they generate electrical currents that can be detected at the surface of the heart. This electrical current is called an electrocardiogram, ECG. In a typical ECG recording, three clearly recognizable waves appear with each heartbeat: the P wave, the QRS complex, and the T wave. The P wave represents atrial depolarization and correlates with atrial muscle contractions or atrial systole. The QRS complex represents ventricular depolarization, and the T wave represents ventricular depolarization. The PR interval is the time interval from atrial contraction to ventricular depolarization. The ST segment represents the plateau phase of the ventricular action potential. A single cardiac cycle includes all of the events associated with one heartbeat. This consists of the atrial systole and diastole, ventricular systole and diastole, and opening and closing of the heart valves. Generally, when the atria contracts, the ventricle relaxes and vice versa. That is, atrial systole corresponds to ventricular diastole and vice versa. The cardiac cycle can be broken down into 5 stages. The cardiac cycle starts with the atrial depolarization, which is the atrial systole. The atrioventricular valves are open throughout this stage. At the end of the atrial systole, the atrioventricular valves close, resulting in the first heart sound, S1. The second stage is the interval between the closing of the atrioventricular valves and the opening of the outflow valves, and is known as the period of isovolumetric contraction. This is the first part of the ventricular systole. At the third stage, the ventricles contract even more, forcing the aortic and pulmonary valves to open. After opening of the outflow valves, there is pumping of blood from the ventricles to the aorta and the pulmonary arteries, and this stage is known as ventricular ejection stage. At the end of ventricular ejection, the aortic and pulmonary valves close, resulting in the second heart sound, S2. At the third stage, the ventricles then begin to relax in order to be filled with blood from the atria. The fourth stage is the period of ventricular relaxation between the closure of the outflow valves and the opening of the atrioventricular valves, and is known as the period of isovolumetric relaxation. At the fifth stage, there is opening of the atrioventricular valves and filling of the ventricles with blood. Sometimes a third heart sound, S3, may be heard during this period. The cardiac cycle starts again with contractions of the atria. Each cardiac cycle constitutes a single heartbeat. In a minute, the heart beats about 60 to 100 times, with an average of 82 beats in a typical adult. The volume of blood pumped by the ventricles in each beat is known as the stroke volume, and is about 70 milliliters. Cardiac output is a factor of the stroke volume and the heart rate. Thus, in an average adult, the cardiac output is 82 multiplied by 70, which is 5,740 millimeters per minute. Thus, the heart pumps between 5 to 6 liters of blood per minute in an average adult.